Hi guys, sorry I'm a few minutes late. So here we go. We are about to do a little short and sweet lesson, I'm hoping, on the structure of neurons. So welcome to you guys who have joined me this afternoon. Really nice to see you all. If only. Hope you're enjoying your times back at school. Is that a joy or not? Anyway, moving on. Since talking about the structure of neurons today, so we are talking about the structure of sensory neurons, motor neurons, relay neurons, or intermediate neurons. It's exactly the same. It's exactly the same phrase, and that whole thing of myelination. What on earth that is all about. So let's see if I can get this show on the road. And as always, we are starting with a little bit of context. And I am taking you back for a little nostalgia trip to GCSE. You'll love this. It all seemed so simple then, didn't it? So I am going to remind you of the stimulus to response pathway, because this will put the context for why you are learning about the structures of the different types of neuron in a neuronal pathway. So we have the stimulus. Remember, those are changes in the environment. The fact that it is getting darker as time goes on into the evening, what you can hear, what you can feel, what you can taste, and what you can smell. So those are changes, obviously, in the external environment, and those are picked up by specialized cells in our eyes, ears, nose, mouth, and skin in order to detect those changes in the environment. And the receptor then causes a neuronal impulse or a nerve impulse to travel down the sensory neuron to the CNS, the central nervous system. And the central nervous system is made up of your brain and your spinal cord. The brain particularly is where everything happens. But in the CNS, that is where coordination happens of different stimuli. The brain, particularly if there are multiple stimuli going on, the brain will coordinate those and then cause a nerve impulse to travel down a motor neuron to an effector. An effector is either a muscle or a gland. And when those muscles contract, and those muscles could be skeletal muscles, they could be muscles that are around in the walls of arteries, for example. Or we could be talking glands. Your favorite dinner could be cooking. You can smell it. Your responses is for your salivary glands to start producing saliva. Or if you see somebody chasing you, your leg muscles start working in order to run away from them. For example, brings about the response. I'm just going to go back to the CNS. The brain is the main coordinator in our bodies, but it's worth noting that the spinal cord still has a coordinator role, much simpler than our brain, but we're going to come on to talk about that role in a minute. Okay. Just to say, if you've got any questions as we're going along, do kind of chuck them in the chat and I will keep looking at my second screen to try and answer them as quickly as I possibly can. There is a little bit of delay. I'm kind of saying this about 10 seconds or so before you're hearing it. So just be aware of that. There may be a little bit of delay in the answer. So just want to point out that these stimuli, the obvious one is when there are changes in the external environment that you hear, you see, you touch, you smell, you feel. But notice that they can also be changes in the internal environment. Your brain has receptors to detect things like the temperature of your blood, the pH of your blood, what else? The pressure of your blood, all of those things you're going to learn about in the course of your A-level studies. So changes in the internal environment are picked up by receptors. And of course, the same basic neuronal pathway is used to bring about a response. And I've already said this, so they're detected in receptors. Just notice we've got receptors on the outside of our body. Most of our receptors are around our head, because that's the first thing that kind of goes anywhere to kind of detect what is going on, and obviously in our skin. But we've also, as I've mentioned, got, have I just said detectors? I should have said receptors, apologies. So we've also got receptors inside our bodies that detect those changes inside. And the outside, we're talking about the senses, and the inside, often that is about homeostatic mechanisms. That homeostasis, keeping the internal conditions the same. If our blood's getting too hot, our body brings about a response so that the temperature comes down. If our blood pressure gets too low, our body brings about a response so that the blood pressure increases again. So all those things are going on inside and outside the body. So in terms of the structure of neurons, you need to know about sensory, relay or intermediate, either term will do, and the motor neurons. Now, really fortunately, again, taking you back to your GCSE, you have come across a neuronal pathway that is made up of just these 3 neurons, and just 3 neurons alone. And that is the reflex arc. So I'm going to teach the structure of the neurons kind of in the context of the reflex arc, because at the end of this lesson, we're actually going to put our knowledge of the structure of neurons at A level into that simple reflex arc diagram, which I'm about to show you now. So only 3 neurons in a reflex arc. It is the simplest of neuronal pathways. And really importantly, because it's often a protective mechanism, this reflex arc means that the message is, the impulses go fast from the stimulus to response, is a really fast reaction, and it's involuntary. It's fast because it's only 3 neurons. It's involuntary because it doesn't actually use the brain. It only goes to the spinal cord. Weird, but true. So here is the kind of diagram that you'd have drawn at GCSE. Stimulus detected by little green receptors. Impulse goes down the sensory neuron, crosses synapse number 1, goes down the relay neuron, crosses synapse number 2, goes down the motor neuron to the muscle or the gland, the effector, to bring about a response. Now, just a note on terminology, so just pointing out, only 3 neurons, only 2 synapses. Pretty much the quickest turnaround time that you can get in terms of stimulus to response. Okay, just a quick note on terminology at A level. Up till now, you have called nerve cells nerves. You're now going to call them neurons. And here's the reason, because strictly, now here is my analogy, I am thinking of a packet of dried spaghetti, okay? Hopefully you can all picture the scene, all right? One strand of that dried spaghetti in the packet is a neuron. The whole bunch of dried spaghetti, i.e. many neurons, is the nerve. Now, the bottom line is at A level, you are practically always going to be talking about the neuron and not a nerve. So actually, I'm just going to say to you, get into the habit of writing neuron instead of nerve. It's going to be incredibly rare where it is more accurate to use the term nerve, i.e. you are really talking about a whole bunch of neurons. Most of the time, you're going to be talking about individual neurons. So just be aware of that. Oh, quick plug, my website. Okay, I've got videos and I'm going to show you that I have now pretty much covered the whole of the OCR A syllabus and the AQA syllabus. I've done videos on every single topic to A-level standard, but really clearly showing you telling you what is in your syllabus and what isn't. Every video has exam questions, me going through exam questions at the end, and for you to subscribe for the whole year. Now, I appreciate most of you haven't got a whole year left, but when you see the price, you still might think it's worth it, because it's still half price. On the website, puts up £100, but in the description, there's the code that you use at the checkout in order to get it for half price. And also, if you just need the AS videos, half price. And if finding the money for this is particularly tricky within the family, I'm getting you to earn, you can earn 10% commission on any friend's purchase. So the possibility of even making a profit on your subscription fee, I'm hoping so. And anyway, there is a freebie, how to improve your grade up to an A star. So at the very least, just grab that. But to show you that I have really practically completed everything, this is all of the topics. Notice one column is OCRA, one column is AQA. Pretty much finished all of the topics. I've just got a couple of videos that I need to edit. Very few topics that need to be finished there. And as you can see, a sea of red because I've got so many videos up there since September. Pretty much there on it all. Anyway, let's get back to neurons. And we're going to start with the first neuron in this reflex arc. We're doing it in order. The sensory neuron. Now, here, I've just realized I've got these in the wrong color. That's irritating. These should be green because I've made my receptor green. I forgot to change the color. These are the receptor cells. That should immediately tell you the direction of the response. Because, of course, this is where the impulse is kicked off. The receptors are detecting a stimulus. So, of course, the direction of the impulse is away from the receptors towards what we're going towards here is the CNS and the relay neuron. Hopefully that makes sense. And you will notice that this is a pretty simple diagram, but it is all the detail that you need to know. And so this, not surprisingly, is the cell body. This is where most all of the organelles are, okay? But I want you to notice that these two long extensions of the cell body are continuous with it. These long extensions, which I will tell you now, are the dendron, which brings the impulses to the cell body, and the little tiny ones are called dendrites. And notice here is the axon, and that carries the impulse away from the cell body. The thing I want to point out is the dendrons and the axons. They are continuous with that cell body, and you've got to imagine them accurately. This is why you need to, because it is what you should be doing, is that those two, the dendron and the axon, are cylinders of cytoplasm surrounded by cell surface membrane that then feed into the cell body, which of course is full of cytoplasm too, and all those organelles. But I just want to point out that that's what the axon and the dendron are like. So, worth noting that the cell body is just on the outside of the central nervous system. So if this is going from my eye to the brain, the cell body is kind of just on the outside of the brain. And the axon is what takes the impulse into the brain, into the CNS, and to connect with the relay neurons. And therefore, doesn't it make sense that the axon, in practically every situation of the sensory neuron, is much shorter than the dendron? Because the dendron, if the dendrons are in the receptors, if the dendron's going from my fingers up to the brain, this is the length of the dendron. The cell body is just on the outside of my brain, and then the axon takes it into the brain. The axon is always shorter. Okay, so now we're here on the relay neuron. Thank you for the very nice comments on the chat. Hilly Dib, thanks, love that name. Right, Tang, action potential. Let me do that at the end of the video, because this is simply covering the structure of the neurons. another word for action potential at GCSE up until A level, you would have called an action potential a nerve impulse. It's the electrical signal that kind of wings its way down a neuron to take an electrical message from, for example, the receptors to the brain. So I hope that helps you for a minute. I think, to be honest, defining what an action potential is, let me think about that. I'll answer that at the end. Hi, Maisha. So back to the relay neuron, and I'm suspecting that you can spot the cell body. Funny that, because a diagram of a relay neuron is practically all cell body, for A-level purposes. And these are the dendrites. Notice that the relay neurons don't have long dendrons, those big dendrons. They have just loads of short dendrites. Okay, so we can tell as soon as we know that something is a dendrite, we know the direction of the action potential. And can I just point out that because these are all dendrites, they would all be bringing impulses, nerve impulses, action potentials into the cell body. And the slightly longer, here's the direction of the impulse, the slightly longer extension in the relay neuron, and notice there is only, or should have pointed that out for sensory neurons, only one axon per neuron. Every neuron only has one axon, may have loads of dendrites, yeah, but only one axon. Really useful to know that. So on a relay neuron, the slightly longer extension is going to be the axon, okay? But notice it's short relative to the other neurons. This is really quite short. These relay neurons are what make up our brain and actually the neurons in our spinal cord. These are the ones that are doing all the coordinating between themselves. All right, they are connected. Get your head around this. They are one. Cell body can be connected to thousands, if not hundreds of thousands, of other relay neurons in the brain. Maybe that has just blown your brains, quite possibly. Blows mine a bit, just incredible. Anyway, on to the last neuron, and here we are. This is your classic year 7, start of secondary school, specialized nerve cell is what you would have called it. And now it's getting its proper name. This is a motor neuron. Notice that the sensory neuron had its cell body kind of in the middle of the dendron and the axon. The relay neuron has a much shorter axon than a motor neuron. And a motor neuron is basically a cell body at one end and one extremely long axon. I'll kind of explain why in a minute. So, not surprisingly, here's the cell body. Here are the dendrites. We immediately now know the direction of the impulse. The impulse is coming in to the cell body via the dendrites. These are all dendrites. So let's just spell this out. Here's the direction of the action potential or nerve impulse. And then it goes down the axon to the effector. And here we have a really long axon. And here's the reason why it's a really long axon. The cell body of a motor neuron is in the central nervous system. It's either in the brain or it's in the spinal cord. And that means for a nerve, for the motor neuron to get down to my feet, the axon for that motor neuron has got to be the length of my legs. Okay, so even for me, over 1 meter long. Yeah, so if I've got motor neurons coming from either spinal cord or the brain down to my hands, well, that's pretty much nearly a meter as well. So the axons on motor neurons are kind of stupidly long. Yeah, you know, much longer, hugely, significantly longer than the axons in sensory neurons, because of course the cell body is right close to the CNS. The axons only kind of taking the message into the CNS. Yeah, the motor neuron taking that message, that, well, should say never call it a message at A level, either a nerve impulse or action potential. Taking that nerve impulse away from the CNS to the effectors, either a muscle or a gland, which bring about that response. And yeah, as I've said, the cell body is within the CNS, and it kind of explains why the axon is so long. So, okay, myelination. Here's the last bit. I told you it would be short and sweet. I think this is, I think I'm going to keep this is going to be true. So really useful for you to recognize that myelination only occurs in some, and I think it's about, if I remember rightly, about 30% of sensory neurons and motor neurons. So you can absolutely think of this thing called myelination, which I'm going to explain in a minute clearly, on sensory neurons and motor neurons. Doesn't happen in relay neurons. I hear by tapping my brain, you know, because that's where my relay neurons are. Okay, so myelination is the insulation of, now very often you're talking about the insulation of the axon, but it can also be of the dendron of the sensory neuron. Notice that it only happens in a motor, here we are, here's our motor neuron, it only happens in the axon because these are little dendrites. They don't get myelinated. It's only the long extensions from the cell body that get myelinated. And this is basically like putting the plastic cover around an electrical wire. It stops, well, no, it doesn't necessarily stop short circuits. That isn't quite true. But the analogy holds true. It is an electrical insulation around the axon or the dendron. So this whole thing is called the myelin sheath. Sheath meaning covering. Yeah, so this is the myelin sheath. And myelin is basically saying that it's made up of a fatty substance which therefore acts as an electrical insulator. And here the individual cells that make up the myelin sheath are called schwann cells. If I remember rightly, dear old Dr. Schwann was actually the guy. Now, he either worked out the cell cycle or mitosis, and I really should know, but I can't remember. Pretty key scientist, can I just say? And he's just got a little cell in our nervous system named after him. I feel like he's been hard done by. Anyway, back to the point. And the gaps between those individual Schwann cells are called nodes of Ranvier. Guess the guy who discovered those was French, so that's the proper way to say that, the nodes of Ranvier, and they are literally where there are bare bits of axon or dendron, bare bits of axon or dendron not covered by this fatty myelin sheath, or the Schwann cells, say either. Now, here is a diagram of a Schwann cell. I love these, I think they're really cool. In the middle here is the axon. We're now doing cross-section, transverse section through the axon. So if my arm was the nerve, I'd be chopping right down through it. Cross-section of the axon. And the Schwann cells literally wrap themselves around the axon, or the dendrons, like a Swiss roll. And hopefully it kind of makes sense that every part of this Schwann cell has got, if it's going round the axon, has got cell surface membrane on the inside and cell surface membrane on the outside. And then you have multiple turns of this Swiss roll cell. So hopefully you can see that you get multiple layers of the cell surface membrane, one on top of the other, making that fatty myelin insulating covering of the axon or the dendron, and it electrically insulates them. And now, here we go, now we are redrawing the reflex arc to A-level standard of neuronal structure. So the stimulus hits the receptors, travels down the sensory neuron, past the cell body just on the outside of the central nervous system to the relay neuron, across the synapses, all right, across this one synapse here, across the synapse here, to the motor neuron and to the effector, bringing about that fast involuntary response because it is such a short neuronal pathway. And here is where the CNS is. Now, I've just drawn the spinal cord here because I am drawing a reflex arc, but it shows you the position of the cell bodies in reference to, in context with the central nervous system. The cell body of the sensory neuron, just on the outside of the CNS. The whole relay neuron in the CNS, brain or spinal cord, in this case, spinal cord, because it's a reflex arc. Cell body of the motor neuron in the CNS with the axon, that really long axon, extending out to wherever the effector is. And I'm hoping that that has now clearly explained the structure of, so we're now going on to any questions. So if you've got questions, right, Munib, I'm going to answer your one in a second. I've seen it. Thank you very much. If you've got any questions, start typing. I'm going to do a quick summary. So we've talked about the structure of the sensory, the relay, and the motor neuron. We've talked about myelination, where it happens, about the Schwann cells and the nodes of Ranvier. And finally, hopefully, we've put what we've learned into context and drawn a proper reflex arc. And so that you can really envisage where those cell bodies are with reference to the central nervous system, and hopefully consolidating that knowledge in a simple diagram that you already knew from GCSE. There's my plan. Okay, questions. Tang Tang, I haven't forgotten yours. Okay, I'm loving the heart. Munib, would you be asked to draw neurons or recognize them? Really unlikely to be asked to draw them, except, I mean, I don't think I've ever seen it on an exam question. I'm not saying you could never be asked to draw it, but only to the level of detail that I've drawn them. I mean, really quite simple. Far more likely, and this absolutely happens, is that they will draw one or more of the neurons and you're expected to be able to recognize them because of their different structures. So hopefully that makes sense. And you would also be able to, you would also be expected to be able to label any part of them. So hopefully that helps you. Okay. Oh, Alex, how does the structure of the glia differ from the neurons in the CNS? They're entirely different cells, entirely different. Glia, do you know it comes from the word glue, stick them together? And actually our brain, I read this actually today. Can I remember the numbers? All I can remember is that there are, if you consider that we have got, I think I'm right in saying something stupid like hundreds of millions of relay neurons in our brain. Less than half of the cells in our brain are neurons. We've got that many glial cells, and glial cells, they think that they're almost kind of maintenance men cells. So they kind of clear up debris, clean out the environment of the brain to enable the neurons to function fully and correctly. Which is why, and I don't think it's been entirely proven yet, but there is a connection with glial cells, and I'm 99% sure I'm saying this is right. Glial cells having a deterioration in the quality and number of the glial cells and onset of Alzheimer's. So I think, glial cells are absolutely fundamentally important to the functioning of the brain, the good functioning of the brain. I don't think their absolute mechanism of action has been worked out in full detail. I'm not a neurologist. I'm going to wave that little white flag at you. But I hope that gives you enough of a feel that they're entirely different in their structure and their function to neurons. They don't carry nerve impulses. Hope that helps. John, hi. Oh, you are very. John, that has just made my day. I'm just so thrilled that it actually kind of unwraps biology, and my videos do that and make stuff clear. Thrilled to bits about that. Seriously, thank you for that. I really appreciate it. Glia could be used to treat Alzheimer's, and that could be due to how you said they clear away debris. I think that's right, but I've got a funny feeling that there are now question marks over the true relationship between the deterioration and Alzheimer's. I'm not entirely sure that has all been sorted out, but I'm on shaky territory, if I'm honest. So I hope that has been useful for you. Oh, Tang. Action potential, right? The best and simplest definition of an action potential is that on the membrane of an axon, it ends up with it being. I'm going to be talking about this next week, actually, when I do resting potential. When I'm doing resting potential. So you end up with it positive on the outside of the cell membrane, the membrane of the axon, positive on the outside, negative on the inside. And when an action potential occurs, it reverses that charge. So it becomes negative on the outside and then positive and reverses back again. And that reversal and back again of charge is transmitted section by section, like a Mexican wave traveling down the axon. And that's why we call it an electrical impulse. We call it an electrical signal, because it is a reversal of that charge. Go into far more detail about that and explain that far more clearly and accurately in my video where I talk about action potential and exactly how that happens. But I hope that just gives you a slightly bigger, better response than what I did before. But I think, unless there are any more questions, I think I'm just going to love you and leave you. I've given you a little half-hour top-up lesson. I think your work is done. Until next Tuesday, when I am going to be talking about a little bit about what happens before the action potential occurs, that reversal and back again, and how the membrane actually causes the situation where there is positive charge on the outside of an axon membrane, if this is my cross-section of the outside, positive on the outside and negative on the inside, ready for some kind of stimulus to come along, or let me reword that, the impulse to come along that axon and cause that reversal and back again charge. So looking forward to seeing some of you again next week, and then the week after, very excitingly, I am going to try and go through past exam papers. Don't think I'm going to manage the whole lot, but I'm going to try and work out a way that I can present the answers that I would have given, present the mark scheme, show why I would have got marks and why I wouldn't, so that I can at least talk about a cross-section of exam questions in a paper. It looks like at the moment on my community tab that me doing a paper one from an AQA A-level exam 2018, me going through that one seems to be winning the votes at the moment. But do vote, because it could change. So there's my plan. Thanks a lot to you, and I will see you next week. Bye-bye.